Yeah, the question in the PYQ previous year paper is Erenchyma is prevalent in, that is a question. Where we find Erenchyma? So Erenchyma, it is one of the adaptations of the hydrophytes. We know Erenchyma is present in the hydrophytes and it is useful for the floating of the hydrophytes on the water. So that is one of the main adaptations of the hydrophytes. And we also discussed earlier about the xerophytes. What are the adaptations of the xerophytes? For example, leaves are modified as spines or scale leaves, and phylloclads, cladodes, and water is stored in the form of the mucilage. All these are the adaptations of the xerophytes. And leaves have the sunken stomata. And in hydrophytes, one of the main features is presence of the parenchyma. That means the parenchyma which stores the air, that is called as parenchyma. And we also discussed about wheat, which belongs to the family OAC. And in part B, so in part B, we also discussed about tunica corpus theory. Who proposed tunica corpus theory? So there are so many theories. I will tell you once. Yeah, apical cell theory. Who proposed apical cell theory? Yesterday we discussed Nageli, Hofmeister and Nageli. And histogen theory. It is related to the root apex, proposed by Hansteen. And tunica corpus theory, it is related to shoot apex and proposed by Schmidt. And Carper Kappe theory, it is related to root apex, proposed by Schupp. And French school theory, it is also related to root apex, proposed by Plantefol and Bouvet. So, and also we discussed about the quiescent center theory, which was proposed by Cloves in case of the root apex. Sorry. So here both the French school theory, it is related to shoot apex. At the same time, quiescent center theory is related to root apex. Both are somehow related. Yesterday we discussed. So these two are dormant tissues. Annuinitial, that is called as annuinitial. It is analogous to the quiescent center in the roots. Annuinitial is present in case of the shoot apex, which is dormant, and the quiescent center is present in the root apex, which is also dormant. But it gets activated whenever required. So you must remember all these theories and who proposed all these theories. Okay, in the meristem, in the shoot apex theories and in the root apex theories, all these will come. Okay, so next. So the question is tunica corpus theory was proposed by Schmidt. Okay, and quiescent center theory. Quiescent center, it is a concept I already discussed you about this one. Root apex. Quiescent center is present in case of the root apex. And cork cambium. What is cork cambium? It is a secondary meristem. We already discussed that meristem based on the origin. It is primordial meristem, primary meristem, and secondary meristem 3 times. So secondary meristem is formed during the secondary growth. Okay, secondary growth in the dicots. Okay, dicot stem. So cork cambium is responsible for the formation of the cork. Okay, This is also called as phellogen. Cork cambium, another name for cork cambium is phellogen. And cork is also called as phellum. Okay. So this is the extrastellar secondary growth. So in the dicot stem, secondary growth is divided into two. Intrastellar, that means within the stele, the secondary growth within the stele, and the secondary growth outside the stele, that is called extrastellar secondary growth, where we find the cork cambium. It is also called as phellogen. Cork cambium produces cork, and phellogen, in case of the phellogen, we call it as phellum. So both they are part of the phellodon, okay, and bark, that is a dead tissue, okay. And dendrochronology, what is dendrochronology? In the secondary growth of the dicots, we see that each year there is one annual ring formation. So based on the number of the annual rings, we can predict the age of the tree. That is called as that study of the calculation of the age based on the number of annual rings is called as dendrochronology. And bioplasts, you know what are the bioplasts? Another name for mitochondria. Okay, so we also discussed about the bioplasts. There is a mistake. Bioplasts, the previous name for the mitochondria. Okay, Altman named it as bioplast. Later, Benda renamed it as mitochondria. And the type of stele present in monocot stem, atactostele. The type of stele present in dicot stem is eustele. Okay, and protostele, actinostele, these are present in lycopodium stem. That is a pteridophyte. And the vascular bundles in Dracena and Yucca are, okay, now we come to this one. There are different kinds of the vascular bundles. Radial vascular bundles, collateral vascular bundles, bicollateral vascular bundles, concentric vascular bundles. In the concentric vascular bundle, there are two types, amphivasal and amphicribal. So now we see different kinds of the vascular bundles here. Okay, so here you see radial vascular bundle. Usually we observe these radial vascular bundles in case of the roots, monocot roots and dicot roots. And there are collateral vascular bundles. These two are called as collateral vascular. Here in the radial vascular bundles, phloem and xylem are arranged in different radius, different radii called as, okay, phloem and xylem. Later phloem. These are arranged alternatively, one after one. So that is called as radial. Okay, next here collateral vascular bundles. In the collateral vascular bundles, xylem and phloem. Usually phloem in upward direction and xylem is located below the phloem. That is called as collateral vascular bundles. Okay, both are arranged in the same axis. In case of the radial vascular bundle, xylem and phloem are arranged in different axes. But in case of the collateral vascular bundle, phloem and xylem are arranged in a same axis. Okay, outside we see the phloem, and here inside we see the xylem. Okay, in between the phloem and xylem, if there is no cambium, that is called as closed vascular bundle. That is found in case of the monocot stem. And if there is a cambium, vascular cambium between the phloem and xylem, that vascular bundle is called as open. Both are conjoint, collateral, and here based on the presence of cambium. If the cambium is absent, it is called closed. If the cambium is present between the phloem and xylem, that is called as open. That is observed in case of the dicot stem. So you should remember all this. Okay. closed collateral vascular bundle present in monocot stem, and open collateral vascular bundle present in case of the dicot stem. And there is one more vascular bundle, collateral vascular, that is bicollateral vascular bundle, bicollateral open vascular. Bicollateral means here, both the sides of the xylem there is a phloem, outside and inside. Xylem is in the middle, and at both the sides we find phloem, that is called as bicollateral. And xylem and phloem is separated by vascular cambium here. So usually in case of the Solanaceae family, we can find this Cucurbitaceae and Solanaceae. It is very important. Where we find bicollateral vascular, in which families? Usually this question is very important, because bicollateral vascular bundles are not present in all the families of dicots. Only in Cucurbitaceae, Solanaceae, such families, bicollateral vascular bundles. So this is very important feature. And now we come to concentric vascular bundles. The question related to in a Dracaena and Yucca, you know, they belong to Liliaceae family. Dracaena and Yucca belong to Liliaceae family of monocots. And here we can find concentric vascular bundles. What is concentric vascular bundle? Here, xylem or phloem at the center. Here, in case of the amphivasal vascular bundle, phloem at the center, and it is surrounded by xylem. It is called as amphivasal kind of a vascular bundle that we see in case of the Dracaena and Yucca. And one more thing you should remember is anomalous secondary growth. As we know, we find the secondary growth only in the dicots, because in the monocots there is no cambium, there is no vascular cambium. So due to the lack of vascular cambium, usually there is no secondary growth in case of the monocots. But Dracaena and Yucca are the exception. There we can find different kinds of the vascular bundles called as amphivasal concentric vascular bundles. That means phloem is surrounded by the xylem. Phloem is located at the center, surrounded by the xylem. There we see anomalous secondary growth that you already studied in your second year. Anomalous secondary growth is observed in case of Dracaena and Yucca. And amphicribal concentric vascular bundle. This is also concentric vascular bundle, but here xylem is at the center. Xylem is surrounded by the phloem. Okay, so here you must you might have understood all these things. Different kinds of the vascular bundles. So now the question is, the vascular bundles in Dracaena and Yucca are amphivasal vascular bundles, but there is a mistake here in Telugu translation. It is called as Kendrikruta Nalika Punjalu anta. Telugulo vitni Kendrikruta Nalika Punjalu. Okay, అంటే ఇందులో పోషక కేంద్రస్థ నాళికా పుంజాలు, పోషక కంజాల మధ్యలో ఉండి దాని చుట్టూ దారు ఉంటుంది, కాబట్టి దీన్ని పోషక కేంద్రస్థ నాళికా పుంజాలు అంటారు. వీటిని కేంద్రీకృత నాళికా పుంజాలు అని కూడా అంటారు. ఓకే, నెక్స్ట్, the root is covered by velamen tissue in. So here options are given: orchids, asters, gladiolus, lily. So here orchids, they belong to the family Orchidaceae. These are monocots. Here the importance of orchids are, they are epiphytes. They are also called as epiphytes. Epiphyte means they grow on the top of the trees just for the sake of sunlight, because they love sunlight. They need sunlight for the photosynthesis. Since they are smaller in size, in thick forests, dense forests, they can't get the sunlight in the moisturous part, because they simultaneously they need moisture and also sunlight. So they grow in the dense forest at the top of the and the branches of the trees. They take the shelter. They take the shelter. They are not the parasitic plants. but they require the shelter. Just they depend for the shelter. So they are called as epiphytes. They have two different types of the roots. Okay, clinging roots and velamin roots. These velamin roots have a sponge-like tissue which absorbs the water in the atmosphere. The water in the atmosphere is in the form of the moisture. So velamin tissue, just like a blotting paper, it absorbs moisture from the. So you might have known, we know several orchids. Just we have to spray water on the roots. That's all, and we don't because they don't grow into the soil. I'm also growing at my house some of the orchids. I will show you one. Yeah. So this is the orchid, which is growing on the tree barks. And they have two types of the roots, clinging roots. They stick to the stems of the trees with the help of the clinging root because they should not fall. And there are freely floating roots in the atmosphere, in the air, which absorb the moisture in the atmosphere with the help of the velamin tissue. So those roots are called as velamin roots, velamin tissue. It is white colored tissue, just like a blotting paper. And next, the stomata are observed in sunken pits. Already we discussed in xerophytes there are specific adaptations just to prevent the transpiration, excess transpiration. That means loss of water. They cannot because they cannot lose water. These xerophytic plants, they cannot lose water in the form of the transpiration. That means loss of water through the stomata is called as transpiration. So to prevent the transpiration, the stomata are located in the lower epidermis and also in the pits. So those are called as sunken stomata or sunken pits. That is one of the adaptation to prevent the excess transpiration, because xerophytes, they need stomata for the photosynthesis, for the exchange of gases. They need carbon dioxide for the photosynthesis. So to facilitate the exchange of the gases, they require stomata, but they are located below the leaves. That means at the lower epidermis. Okay, they are restricted to lower epidermis in the pits to prevent the transpiration. It is called so these sunken stomata are present in xerophytes. And the bark-like protective covering found in underground stem seeds. So this is one more question. The bark-like protective covering found in underground. So we know there are so many runners, suckers, okay, comb. So all these are different kinds of the underground stem modifications. So here, those underground stem modifications are protected by a specific tissue, covering tissue, that is called as polyderm. So you remember this term polyderm. It protects the underground modified stems. Okay, and the radial conduction of water after secondary growth is primary by. So you know, after secondary growth, we already discussed secondary growth usually occurs. It takes place in case of the dicot stem. Okay, in the dicot stem or in the root, dicot root, there are some specific structures. Those are called as medullary rays. You might know. Yeah, this is, you see in the secondary growth. Here you see the medulla or pith. At the center of the stem, we find medulla or pith, and there is a connection between the outer cortex to the medulla in the stele. That connection is in the form of the medullary rays. So they are useful for the water transportation, radial water transportation. Okay, radial. That means It is not a straight one. Perpendicular to the axis. Perpendicular to the axis. That is called as radial transportation of water. Takes place by medullary rays. Okay. So next. The ovule resembling the horseshoe shape. So you already studied in embryology different kinds of the ovules that we see here. So there are different kinds of the ovules. Okay. And this is the arthropous ovule. It is the straight one. Hemitropous ovule. It is perpendicular. That means 90 degrees. It is bent 90 degrees in the angle of 90 degrees. That is called as hemitropous. So you must read the examples also. Polygonum. Arthropous ovule. Example is polygonum. Cycas. And hemitropous. This is one of the primitive feature. Arthropous, presence of the arthropous ovule is a primitive feature. Usually we find in the gymnosperms also and in the lower angiosperms. And hemitropous ovule, that is 90 degrees. It is bent 90 degrees. Ranunculus, hemitropous. And campylotropous ovule. Usually we find this ovule, that is a characteristic feature in case of the Leguminosae, Fabaceae. Okay, campylotropous. That means it is bent little more than 90 degrees. And you see anatropous ovule. That means inverted ovule. Ovule body is inverted 180 degrees. The angle is 180 degrees. And horseshoe shaped ovule. Here, here it is just like anatropous ovule, but here what we observe is the embryo sac is horseshoe shaped. That means U shaped. That is and one more thing is sarcinotropous ovule. In case of the Opuntia, we find this ovule, sarcinotropous ovule, horseshoe shaped ovule, embryo sac. So you see within the ovule there is embryo sac. It is U shaped or horseshoe shaped. So that is the answer for this question. Amphitropous ovule, where we find the embryo sac. It is not the ovule resembling the horseshoe shape. It is the embryo sac in the horseshoe shape. Okay, that is called as amphitropous ovule. So you must read different types of the ovules and their angles, bending angles, and their examples, their special features. And the next question: the entering of the pollen tube into the ovule through the middle part, piercing the integument, is called. Okay. So how the during the fertilization, after pollination, the pollen on the stigma of the ovary, it germinates and it forms pollen tube, and it grows through the style and enters into the ovary, and it reaches the ovule. And how it enters into the ovule? Okay, how it enters into the ovule? If it enters through the micropyle, that is called as porogamy. Okay, if it enters through the chalaza, it is called as chalazogamy. And if it enters through the integuments of the ovule, that is called as mesogamy. So syngamy is a different term. It is not related to pollen tube entry. Syngamy, what is syngamy? You must know. Okay, the fusion between the one of the male gametes and the female gamete is called as, or egg cell is called as syngamy. Okay, so it is not related to this question. Mesogamy is the answer. So you may see so somewhere I have kept the diagram for you. Yeah, so here you see this is the ovule and here pollen tube enters into the embryo sac through the micropyle. That is a pore, is called as porogamy. And here you see this is the ovule and opposite to the micropyle there is a chalaja or kalaja. So pollen tube enters through the kalaja or chalaja. It is called as kalajogam or chalajogam. And next here you see this is the answer for this question. The pollen tube enters into the embryo sac through the integuments from the middle. It is called as mesogamy. So the answer is mesogamy. And now endosperm. The question is related to endosperm. Endosperm is haploid in. Where we find endosperm in the haploid form. So I will show you one thing. It is very important for you. Yeah. You see, in the structure of the ovule, just now we have seen it, ovule structure. See, here this is the micropyle, and there is a nucellus. It is surrounded by two integuments. Usually, in case of the dicots, in the polypetalae members, we see two integuments. So they are called as bitegmic, and in case of the gamopetalae, usually we see only one integument, that is called unitegmic. Okay, and one more thing is here, there is a nucellus, and within the nucellus there is a female gametophyte. It is also called as embryo sac, and within the embryo sac we find towards the micropyle there is egg apparatus, two synergids, and at the center there is a egg cell. It is called egg apparatus. At the center we find secondary nucleus or central cell. It contains two polar nuclei, and towards the chalaja in the embryo sac there are three cells. It is called as antipodal cells. Okay, you know all this. So here what we see, the integuments of the ovule are always diploid. Always diploid. Before fertilization and after fertilization, so they are diploid. And after the fertilization, these integuments, they form the seed coat. The outer integument is called as testa, and inner integument is called as tegumen after the fertilization. So this ovule after fertilization becomes a seed. Okay, once ovule is fertilized, it develops into a seed. Okay, these integuments, they become seed coats. Outer one is testa, inner one is tegumen. And after the this nucellus, it is a nutrition. It is the purpose of the nucellus is to provide the nutrition to the growing embryo sac. So it becomes, the nucellus is always diploid, and it is also named as perisperm. After the fertilization in the seed, the leftover nucellus in the seed after fertilization is called as perisperm. It is diploid. Before fertilization diploid, and after fertilization also diploid. After fertilization, nucellus is called a perisperm. Okay, and here you see there are three cells in the egg apparatus. The middle one is called egg cell. It participates in the sexual reproduction. That is called as syngamy. So we already discussed through the pollen tube, two male nuclei or sperm cells enter into the embryo sac. One of the male gamete or male nucleus or sperm cell, it unites with the egg cell. This is called as syngamy. Okay, and this is the first fertilization. And after the syngamy or fertilization, egg cell becomes the zygote, diploid zygote, and it develops into a diploid embryo, an embryo. Okay, and next there are two polar nuclei here in the central cell or secondary nucleus. It is called as secondary nucleus. It is already diploid, and when another male nucleus or male gamete enters into the embryo sac, it fertilizes this secondary nucleus and it becomes diploid. This process is called as double fertilization or triple fusion. Okay, and also you should remember who discovered this. Syngamy was discovered by Strasburger. And triple fusion or double fertilization was discovered by Navashi. So these are very important. Okay, Strasburger, Singami by Strasburger, and double fertilization. The same phenomenon is explained in two different terms. Okay, triple fusion because it involves the fusion of three nuclei, two polar nuclei and one male nucleus. So it is called triple fusion. And double fertilization, it is a second part. So it is called. So it forms triploid primary endosperm nucleus after the double fertilization. This is also called as PEN, primary endosperm nucleus. It develops into endosperm. Okay, so endosperm is always triploid in case of the angiosperms, but it is haploid in case of the gymnosperms. So you should remember this one. So endosperm is haploid in case of the gymnosperms, but triploid in case of the angiosperms, which include dicots and monocots. In monocots, it is triploid. In dicots also, it is triploid. But in case of the gymnosperms, it is always haploid, and there is no endosperm in pteridophytes. And the next question: the explant for anther culture is taken from. So for anther culture, explant, the tissue which we use in the tissue culture from the mother plant is called as explant. Okay, so explant is inoculated on the tissue culture medium. So we always use for the pollen culture, for anther culture, we use the unopened buds because we have to take the pollen. There is a pollination from one plant to another plant. So to make sure that we are using the explant from the genuine plant, we have to use the unopened buds. If we use the opened buds, some pollen from other plants also may fall on that flower. So we always use the unopened buds for the pollen culture or anther culture. And next, pollination by insects is called. So here, pollination, myrmecophily. What is a myrmecophily? The vector. The pollination with help of the ants is called as myrmecophily. In some of the plants, like Acacia spirocephala, it shows myrmecophily. There is a friendship between the ants and this plant. Okay, so these ants help in the pollination in case of the Acacia spirocephala plant. So it is called a myrmecophily. Anemophily, the pollination with the help of the wind is called as anemophily. Entomophily, the pollination with the help of the insects mediated, because most of these times we see butterflies, bumblebees, all these are insects. They fall under the category insects. So it is called as entomophily. Ornithophily means the pollination with the help of the birds. Erythrina. Okay, in Erythrina plant we see ornithophily. Okay, so here I want to show you one diagram here. Yeah, so abiotic pollinators and biotic pollinators. Abiotic pollinators means wind mediated pollination, anemophily. Water mediated pollination is called hydrophily, and biotic pollinator, insect mediated pollination is called entomophily. Bird mediated pollination is called ornithophily, and there is bat mediated pollination also, chiropterophily. So you must remember all these names. Chiropterophily is bat mediated, bats. Okay, they mediate the pollination. So we have lost one more minute. And last question here, endothelium is the layer of specialized cells. Where we find endothelium? Usually we find endothelium surrounding the embryo sac in the ovule. There is a thin layer surrounding the embryo sac that is called as endothelium. It is very important question because usually it is not discussed in the normal materials and textbooks. Okay, so you should remember this one. And the endosperm which is irregularly ridged and furrowed is called as. There are three types of the endosperms: helobial endosperm, cellular endosperm, nucleate endosperm. Last one is ruminate endosperm. So this is the answer in Anonaceae family, and in Myristicaceae family, we find the wave-shaped, ridged, furrowed endosperm, that is called